voice is clear?
Yes, sir.
Okay. Let me just make a bit modification in that so that it is more audible. Okay, now I think it will be a bit better, huh?
Yes, sir.
Okay then. So good afternoon and let us start because in morning there was some issues with so that we could not start it. Can you see the slide, Samara?
Yes, sir.
Okay. And I hope the recording is now automatically on. I do not have to do it. Yes, Samara?
Yes, sir.
Someone is there from the recording section?
Yes, sir.
Okay, so that means they are doing it automatically.
Yes, sir.
Right, so in our previous class, we largely discussed about agrogel electrophoresis and then we started discussing about page. What is full form of page?
Polyacrylamide gel electrophoresis.
Okay, good. And polyacrylamide gel electrophoresis is largely used for protein
Proteins. Yes, sir.
separation. Okay, although it
Yes,
can
sir.
be used for nucleic acid as well, like when we are doing sequencing. In that case, we have to separate like one nucleotide each. So in that case, we use page for
Page.
DNA
Okay,
as
sir.
well, but largely it is used for protein. So there are two kind of page: native page and non-native page, right? So
Yes,
native
sir.
page is called non-denaturing, in which we use the non-denaturing agent. I think this
Yes,
part
sir.
we discussed in previous class. I just want to just give you an overview. Okay.
Yes, sir.
And in non-native page, we have the SDS page. So what is difference between native page and non-native page?
Sir, in native page, in native page is used when we have to make the protein intact,
Mhm.
and in non-native page we can denature the protein.
Okay. So why we do denaturation of protein when we want to? We can have the native, we can have the
Sir,
native.
in order to study the structure of a protein, we have to
Yes, yes, yes.
denature
I
it.
have told you, if you remember,
Yes,
that
sir.
separating a protein in its native form is very, very difficult. Very
Yes,
difficult.
sir.
But separating protein in denatured form is easy.
Yes,
So
sir.
we generally do non-native page for structural analysis, and if we have to go for activity analysis, in that case we have to go for the native page.
Native page. Yes, sir.
Hope it is clear, huh? And I think we
Yes,
have
sir.
discussed about the different component of SDS and how it is done, and I think it was over up to here. Okay.
Yes, sir.
Now we have to discuss about the native page. And native page, I have told you, I think we have started discussing. So native page is otherwise also known as 2D gel electrophoresis. 2D. So in 2D gel electrophoresis, other way it is correct, that in 2D gel electrophoresis, one is native and another is non-native, right? So
Right,
in
sir.
first dimension, we do isoelectrofocusing. So two dimension means when the separation is done on two concepts. Like in SDS page, what is the principle of separation of molecule? In SDS
Sir,
page, what is the principle of separation of molecule of proteins?
sir, in that case, we add the SDS molecule on the protein on the polypeptide chain, and it will impart the
That's okay.
negative.
But how it is, like, what is the principle of separation? What is the physical concept of separation?
So it is separated by determining the mass to charge ratio.
Yes, yes, good. So not mass to charge ratio precisely, you can say. It is based on the size, based on the
Yes, sir.
mass, based on the
Mass.
mass. Separation is largely based on the mass,
Mass.
molecular weight, or we can say mass.
Yes, sir.
In case of 2D electrophoresis, we run two type of electrophoresis. One is called isoelectrofocusing, in which the separation is according to isoelectric points, pi, point of isoelectricity. I'll discuss this part. And in second dimension, the separation is based on molecular weight
Molecular weight. Okay,
in
sir.
SDS space. So the Second dimension in 2D is SDS page, and the first dimension is isoelectrophoresis.
Focusing. Yes, sir.
Right. So
Right, sir.
how we start actually, that we take like proteins can be negatively charged also, positively charged also, and zwittronic also.
Yes, sir.
And the charge on the protein, it depends largely on the pH. So at one pH, the protein may be positively charged. At another pH, it will be neutral, and another pH, it will be positively charged. Same protein, right? So the
Okay, sir.
charge on the protein is a function of pH on which the protein is lying, right? You must
Okay, sir. Yes, sir.
you will remember, I think, that there are two uh, deprotonation group in the protein. One is the ammonium group NH4 plus. When it is NH4 plus, we say that the amino group is protonated. So when our amino group is protonated, then it is NH4 plus.
Yes, sir.
We have the second functional group of amino acid. What is the second functional group? The second functional group is carboxylic group.
Yes, sir.
Carboxylic group is fully protonated. Then it is in which form? COOH form.
COH form. Yes, sir.
Now, is it like charged or uncharged?
Sir, it's uncharged in
It is
COH
uncharged.
form.
Very good, very good. It is uncharged. Now, when it will be deprotonated, it will be deprotonated and it will be converted into carboxylate
CO
ion,
negative.
C
Yes, sir.
double bond COO
Minus.
minus, right?
Yes, sir. Yes, sir.
So, a protein can be, when a protein is fully protonated, it is positively charged,
Mm-hmm.
and when a protein is fully deprotonated, then it is negative.
Negatively charged.
So, like one deprotonation will be from the NH4 plus group, and it will be converted into NH3, and NH3 is what? Is it positive or is it neutral or charged?
Sir, NH3 is positive.
No, no. NH3 is again neutral. NH4 plus is positive.
Yes, sir. Yes, sir.
Okay, so again it is neutral, and
Mm-hmm.
then one proton will be lost from the carboxylic
Mm-hmm.
group, and then it will be converted into carboxylate ion CO minus.
Yes, sir.
Now, since a protein is made up of many amino acids, it is a polypeptide. So one amino acid may have different pi, another amino acid may have different pi. What is pi? Pi is, you can write down, pi is the point of isoelectricity, the point at which net charge on the amino acid is zero. Means the NH4 plus group is fully protonated. That means it is NH4 plus, it is heavy carrying a positively charge. And the carboxylic group COOH group is deprotonated. So it is COO minus. So that means
Yes,
the
sir.
amino acid is carrying one positive charge and one
And one negative
negative charge.
charge.
And
Zero.
net charge is
It's
zero.
neutral.
So when the net charge is zero, actually it is not neutral. That's why it is called zwittronic. It is not called neutral. It is called
Mm-hmm.
zwittronic. Means
Zootronic.
the protein or the amino acid is having both the charge. But
Mm-hmm. Yes, sir.
there is equal charge. Means if there is one
Mm-hmm.
positive, there is one negative. If there are two positive, there are two negative. If there are three
Two
positive, there
negative.
are
Okay,
three
sir.
negative. Like this. So the whole protein, it becomes zwittronic. And if the protein is zwittronic, will it have electrophoretic mobility?
No, sir.
It will not have electrophoretic mobility. Why? Because the net charge is zero. The
Zero.
requirement is
Yes, sir.
for a protein to move under electrophoresis, it must possess
Hmm.
a net charge.
B charge.
Okay?
Yes, sir.
Right? So, in 2D electrophoresis,
Okay, sir.
we explored this idea that the charge on a protein is a function of the pH. So, what we did, we made a system in which we took a plastic strip, and on that strip, we we applied gel. Again, it was polyacrylamide gel, but that polyacrylamide gel was having pH gradient. That was having pH gradient,
Ph
right? You got
gradient.
it? pH gradient
Yes, sir.
means like there may be range of pH, say 4 to 7, 4 to
Yes, sir.
7. So
7.
that means one end is having 4 pH, another end is having
Mm.
7 pH,
7
or
pH.
maybe say 4 to 9, or maybe say 4 to 12. So like, or say 3 to 12.
Okay, sir.
Like this, we may have broad range of pH or we have narrow range of pH. So how it is done? Like simply suppose I have to make a gradient. So I will make gradient gel, and the gel is having ampholites which are having different pH. So one end is having say 4 pH, another
Mm.
end is having say 7 pH. And
7 pH.
on
Okay,
this
sir.
strip, this is called IPG strip. This is called IPG strip. Okay,
IPG strip.
IPG strip, or
Okay,
we
sir.
call it. It is called isoelectrofocusing. Now what we do, we apply protein on the whole length of this strip, and then it is kept under a power supply. Okay, so and then we allow it to stay in power supply at very low voltage for say 6 hours or overnight. So what will happen, that the protein, since the charge on the protein is a function of pH, now if a protein here, it is having negative charge, now gradually it will move towards the positive electrode. If a protein here it
Positive
is
charge.
having
Yes, sir.
negative charge, positive charge, gradually it will move towards the negative electrode, right?
Negative charge.
Now, while
Yes, sir.
moving down the pH gradient, it will come across a pH where it became zwittronic.
Deuteronic.
Now,
Okay, sir.
wherever it will become zwittronic, its electrophoretic mobility will become
0.
zero, and it is fixed here. It is
Yes, sir.
focused here. We call it focused here. That's why it is called isoelectrofocusing.
Okay, sir.
Isoelectrofocusing means
Focusing.
if at a point where the protein it becomes zwittronic, it will stay here. Right? Like now it is fixed here. It is staying here. Like whatever length of duration you run this plate,
Okay, sir.
but this protein will not move. So overnight, what happens? Like these proteins, they got separated based on their what? Based on their
Hello,
PI.
sir.
Yes.
Hello, excuse me.
Yes. Can you hear me?
Hello. Hello, sir. I have to ask a question, sir. Which slide? Yes, sir. Yes, sir. Sir,
Hmm?
which slide you are showing?
I'm showing it is written two-dimensional electrophoresis.
Uh,
You cannot see that?
no, sir. I can only see the native page isoelectrophoresis.
You should have told me,
Yeah,
na?
now, sir, I can see it. Okay, sir.
Now, can
Thank
you
you.
see that? Huh? Just a second.
Hmm.
Huh?
Yes, sir.
Hmm. Now, can you see that? You keep on telling me, na? Now, can you see it? Can you see it
Yes,
now?
sir. I can see it now. The two-dimensional electrophoresis.
Okay. So you can see this strip. This is IPG strip, and you
Yes, sir.
can see the proteins which
Yes,
are
sir.
on the this thing, and the proteins are separated based on their
Yes, sir.
PI. Okay, right? So like suppose this protein is having a PI
Mm,
of say 6, so it will be here. PI of 4, it will be here. PI of 7, so it will be here. Like this. Okay, clear?
Okay, sir.
Just a
Okay,
second,
sir.
please. Just a second. Just a second. Just a second. Yes, Samra, can you hear me?
Yes, sir.
Okay, wait. I'll just share the slide again. Can you see the slide? Yes, Samara, can you see the slide?
Yes, sir.
Okay, so in first dimension the protein is separated based on their pi, based on
Sir,
their pi. Okay.
PI.
Okay, pi is isoelectric point, the
Okay, sir.
pH at which the net charge on the
Yes, sir.
protein becomes zero.
0. Okay, sir.
Then this strip is loaded on a SDS page, and then it is separated based on
Their
what?
size.
Based on size or molecular weight, right? So
Their
the first
size.
separation is based on pi,
Yes, sir.
and second separation is based on molecular weight, right?
Their size. Molecular weight. Yes, sir.
Molecular weight. So that's why it is called two-dimensional electrophoresis. One dimension is for pi based separation, another dimension is for molecular weight based separation, right?
Molecular weight.
And rest is same, like
Right, sir.
the way whatever way we want,
Mm-hmm.
accordingly we can we can stain it with the Comazzi blue, or we can stain it with silver stain, that what we discussed in our previous class. Okay. And what is the use of this
Yes,
technique?
sir.
It is one of the
Okay,
best
sir.
technique today for doing like disease diagnostics. Like you have, this is condition A or condition B. Means suppose condition A is normal. These are normal human cell, and you can say these are the variegated human cells. Now you take out protein from them, both the cells. Run.
Sir,
Yes,
sir,
I have
sir,
changed
have you
slide.
changed the
I've
slide?
gone to the next slide.
Like,
You cannot see it.
sir, I cannot see that slide.
Okay, okay. So now onwards, I will select
No,
next.
sir.
Now you can see it, I think. Right? Can you see it?
Yes, sir.
Okay. So I think the slide is not moving. I do not know why. So I think I'll just keep it like this only. Yeah. Now you can see it. I think it will be little bit
Okay, sir.
smaller view, but I think you can see it. Is it okay now?
Yes, sir, I can see that. Okay, sir. Yes, sir. It's okay.
Because it will be little bit smaller view, because it is not in the
No, sir, it's
slide
okay. I can
view
see
mode.
that.
Okay. So, like we can take protein from two sources. One is a normal cell, another is a diseased cell. Then first do separation based on isoelectrofocusing, then run SDS page, and then you can compare the protein from both the cells, and then you can identify that in diseased condition some new proteins have either appeared, or some expression of some protein has changed, right? And then these proteins can be submitted for mass analysis for identification of these proteins. So this 2D electrophoresis is one of the very what should I say, one of the very advanced and very widely used technique for proteomics analysis, whole proteomics analysis. Okay, I hope
Okay,
you
sir.
know what is a proteome.
Yes, sir.
So proteome is what?
Sir, proteome is defined as all the proteins that are produced by a genome.
Yes, good. Total protein makeup of an
Hmm.
organism is called a
Proteome.
proteome. Okay, so the proteome is like it is. We can have the idea of whole proteomes, and that whole proteome can be it can be analyzed, and we can find out the variegated proteins or the unusual proteins. Okay, you
Okay, sir.
can see the next slide.
Yes, sir, I
Can
can
you
see the
see the
next slide.
next slide?
Yes, sir.
Okay, so the next slide is like first we prepare sample, then we have then we do isoelectrofocusing, that is first dimension, then we run SDS page, second dimension, then we visualize the protein spots by staining, and then we identify those proteins. Okay.
Okay, sir.
Okay, so this is in detail about that. So you can just read it, and this is the equipment you can see. So this you can see, that is, this is the like the strip.
Strip.
And you see
Okay.
that we apply the sample here, and you can see that the protein, this is positively charged end and this is negatively charged end. And you can see, like, this protein, it is having a pi of 5.1. So earlier, if you see, this protein was like, so like three proteins, so pi 5.1, pi 6.4, and pi it is
8.6.
8.6. So like this, you see that there is bidirectional movement of the protein. But in SDS page, if you see, there is unidirectional. Now this
Unidirectional.
is kept on SDS page, and then you can see, and then we can find out that some of the proteins, they are different. Okay.
Mm.
Now
Okay, sir.
these different protein in disease condition, these are different. So these different proteins, they can be analyzed by mass, and we can actually identify the proteins, which are those proteins, and then we can find out their prediction if it is already their function if it is already reported, and if not, okay, so we can just find out the structure of the protein, and then we find out the function of the proteins. Okay.
Okay, sir.
So this is one of the very versatile technique available with us, and we can use this technique for, these are some of the experiment which actually I am already working in proteomics. So these some of the experiments from our uh, our experiments. Okay.
Okay,
Then
sir.
the next technique is capillary electrophoresis. Next technique is capillary
Capillary electrophoresis.
so far we have seen agarose gel electrophoresis, page, and then now we have the uh,
That
uh,
two-dimensional
two
SDS page.
dimensional SDS two dimensional, that is called isoelectrofocusing,
Mm.
and now
Yes,
we
sir.
have the capillary electrophoresis. So what do you mean by capillary? Capillary means a very fine tube.
Mm. Yes,
Capillary
sir.
means a very fine tube.
Very fine tube.
Now, what happens actually when we are separating protein? Have you run some SDS page gel in your MSc? SDS gel in your MSc?
No, sir. I have worked on the agarose gel.
Agro gel, you might have heard, work,
Yes, sir. Yes,
So
sir.
you might have seen that the in agro gel, the gel is always submerged in water, in buffer.
Buffer. Yes, sir.
So if the buffer level is low, then what you believe what will happen? What will happen?
Sir, if the buffer level is low, then the DNA will not be able to move properly in the gel.
Why? If I am saying your gel is half dipped, not fully dipped, it
Hmm?
is not fully submerged, it is half submerged, then what will happen? Actually, what will happen, that your, like when we are pulling a molecule from inside the gel, then the molecule is experiencing a lot of friction.
Okay, sir.
And due to that friction, there is generation of high heat. So the gel
Okay.
is always dipped in buffer, so that the heat generated due to friction is transferred to the buffer.
Buffer.
And
Okay,
you
sir.
might have seen that whenever we do electrophoresis, it is recommended that it should be done at around 20 degrees centigrade.
Yes, sir.
Like, it must be done in AC. If
Yes,
you
sir.
do it in 35, 40 degrees centigrade, and if you do it in 20 degrees centigrade, there will be change in the results. Okay?
Okay, sir.
Because friction is a parameter of temperature.
Temperature. Yes, sir.
So friction, what happens? Another thing is that when, like you might have seen, that if you pull the protein fast, then friction is more. If you pull the protein slow, then
Mm.
friction is less.
Is
That
less. Yes,
means
sir.
friction is again a parameter of the strength of the applied electric field. So if the strength of the applied electric field is more, we will try to pull the molecule fast, and we will try to pull the molecule fast, then it will experience
More
more
friction will be there.
friction and more heat will be generated. Okay.
Yes, sir.
So in SDS page, generally it takes say 3 hours or 4 hours for separation of a protein. Now, what if I want to separate the protein in 15-20 minutes? Is it possible? Can I run SDS page in 15-20 minutes?
No, sir.
It won't. Why it won't? Because SDS page uses low electricity what we have, right? We have
Right, sir.
low electricity, right?
Mm-hmm.
So one idea came that instead of running the gel for 3 4 hours, if I can run the gel in 15 minutes, how it can be done? So it can be done by increasing the what strength of the applied
Strength.
electric field.
Electric field. Yes, sir.
So instead of like you might be remembering that generally we run SDS page in separating gel at around say 60 or 70 volt,
Mm-hmm.
60
Yes,
or
sir.
70 volt.
70 volt.
If I make it say 1000 volt, then what will happen? We can pull the protein fast,
Mm-hmm.
but very high friction will be generated and very high heat will be generated.
Mm-hmm.
And the plates between which we have casted the gel in a page, these plates will break.
Okay, sir.
These plates will break. So if we want to do the separation fast, then heat dissemination must be very efficient,
Okay, sir.
right?
Right, sir.
So keeping this in question in consideration, we invented capillary electrophoresis, in which what we have that the electrophoresis is conducted in a fused silica capillary, means it is a very fine capillary, and around that capillary, which is around 10 to 100 micrometer in diameter and around 40 to 100 centimeter long, and then what we do, we apply a high cooling device, like this capillary is kept in a very high cooling device, so that whenever we will be pulling the protein very rapidly, then whatever amount of heat is generated, it is immediately transferred to the coolant. Okay, you got it?
Okay, sir. Oh, yes, sir.
And then we apply very high voltage. You see 5 kilovolt to 30 kilovolt. Huh?
Mm. Yes,
5
sir.
kilovolt means we can say around 5000 volt to 30,000 volt.
30,000
Can you
volt. Yes,
imagine?
sir.
Huh? Such a
Mm.
high
Yes, sir.
voltage. Huh? And
Yes, sir.
the sample injection is also automatic and detection is also automatic. Now you
Automatic.
see it is done like this, that we have a fine capillary. Now this capillary is having a cooling device around it, and then you inject the sample here. So like a moving boundary electrophoresis, similar kind of system we made, and then we have a very strong power supply. We have an automatic detector, so whatever compound is coming here, it is detected, right? So
Okay,
this
sir.
is a generic design for a capillary electrophoresis. Now I'm sure I'll show you just one video. You can see like all proteins are loaded from here. I think I do not know whether I think it will only run in this mode. Can you see it now?
Yes, sir.
So you can see. I just repeat it again. Now can you see?
Mhm. No, sir.
Now you cannot see. Now you can see. I think. Can you see the components
No.
moving? It's actually
No,
animation.
no, sir. No, sir.
You cannot see it. Now can you see one slide in which we have some colored analytes at cathode?
Yes, sir.
Can you see
Yes,
my
sir.
cursor? Okay.
Yes, sir.
Now again, can you see the
Mm.
same slide?
Yes, sir.
Now can you see the slide where these analytes are going from cathode to anode?
No, sir.
Okay, wait. Possibly I need to share it again, then only it will be visible. Okay, now is it visible?
Yes,
Yes,
sir.
Samra? And now also?
Yes, sir. It's so the slide is visible, but the thing that you are saying is moving. I can see that. That.
You cannot see that, huh? Okay. But I think it is on slideshow
No, sir.
mode. You cannot see the components moving and development of a chromatogram.
No, sir.
Okay. Wait. Okay. Now you can see.
Yeah, no, yeah.
Okay.
Yes, sir. I
Anyhow,
can
you
see the
can see
final
that.
one.
You can see the final one. Now you can see the cannot see the animation. So these molecule
Yes, sir.
earlier you might have seen these were here. They travel up to here,
Yes,
and
sir.
then we have a digital recording of the data, just like we do in chromatography.
Hmm.
And here you see that this they are moving through this
Okay,
fine
sir.
capillary, and it is done at a very high voltage. Okay, you got it. So that means using this very high voltage,
Okay, sir.
we can
Yes, sir.
separate the components in a very rapid time. Okay, so this technique is called capillary electrophoresis.
Okay, sir.
So, and we use online
Capillary
detectors, like
electrophoresis.
detectors similar to those used in GC and HPLC can be used, and majority of the instrument have UV visible detector, right? Alternatively, for quantitative analysis, we can have detector like fluorescence, and mass can also be used very frequently, right? And then we
Okay,
get
sir.
a
Right, sir.
electropherogram.
Parogram.
Like in case of HPLC, what we were getting in HPLC, what we were getting,
Chromatogram.
we were getting a chromatogram. Here, what
Chromatogram.
we are getting a
Yes, sir.
electropherogram, which is like a chromatogram.
Electropherogram.
Okay, because it is a plot of
Okay, sir.
time versus the components. Okay, the
Okay, okay,
signal
sir.
on y-axis. So it is mostly like the milli-absorbance signal in case if we are using the UV visible detector, or the mass data if we are using the what do you call it, if we are using the mass detector. So we have the mass on the
Mm-hmm.
y-axis if we are using the mass detector. Okay.
Mass
So
detector.
I
Okay,
think
sir.
with that we have finished up electrophoresis part. So if you have any question, please ask me. Any question if you have.
Sir, yes, sir.
Mhm.
Sir, 1 minute, please.
Yes, yes, no problem. Take your own time.
Sir,
Yes, Samara.
hello.
Yes.
Yes, sir. Can you please re-explain me discontinuous SDS page?
Which one?
Sir, discontinuous SDS page.
Okay, okay. I think I have not shown you in my slide this part.
Yes, sir. You have explained me in the last class, but
Mhm.
due to poor connection, I was not able to get the whole thing.
Okay, SDS page you want, eh? Wait, wait. Let me just go back. Yes, Samara, can you see the slide?
Yes, sir.
Okay. So this is SDS page, and SDS page is a non-native electrophoresis. Okay.
Yes, sir.
And what we do, we treat the protein with SDS. Can you see the next slide?
Yes, sir.
So it is sodium dodecyl sulfate, which is having sodium. Then sodium is ionically bonded to sulfate, and sulfate is covalently bonded
Yes,
to
sir.
hydrophobic chain. Okay, 12
Right.
carbon
Chain.
hydrophobic
Yes, sir.
dodecyl.
Hydrophobic
Okay,
chain.
is it clear? Now,
Yes,
when
sir.
we put it in water, then what happens? Then since this sulfate and sodium are under the ionic bond, and ionic bonds are like they got dissociated when we put the salt in water. So this bond get like it got dissociated, and due to dissociation of the bond, we have another molecule that is called dodecyl sulfate. Now, what will be the charge on the dodecyl sulfate? Charge on
Sir,
dodecyl
negative
sulfate
charge.
is negative charge, right? Now, this negatively charged
Right, sir.
dodecyl sulfate, it when we mix with a globular protein, then it will make this protein linear, so that separation of that protein becomes easy.
Yes,
Separation
sir.
of that protein become easy,
Easy.
and second thing is that
Easy.
it gives a universal negative charge to it, right? So initially, like you see, that protein was also having somewhere positive charge, but after
Okay, sir.
treating with SDS, it will be now universally negatively charged. Okay, because
Negative charge.
the negative
Yes, sir.
charge of dodecyl sulfate, it will mask the protein, and now the protein will be negatively charged. Okay, so
Negatively charged.
either we can have continuous SDS page where the gel is like having same porosity, or we can have discontinuous SDS page where we have a stacking gel and a resolving gel. Okay, so what happens actually in the gel, I tell you that when you are making a polyacrylamide gel, you see here in discontinuous SDS page that we have a top gel that is called stacking gel, which is 4% acrylamide, and you have a separating gel, bottom gel, which is from 5 to 15%. Generally, we use 10% or 12% of acrylamide, right? So what happens actually
Okay, sir.
that suppose this one is the top one is your stacking gel, right? And the bottom is your resolving gel. So what happens that when
Okay,
we
sir.
load this sample on the in this well, then generally what happens that
Yes,
when
sir.
we are loading the sample in the well, then sometime the well may not be very uniform. What happens? Suppose you are casting the gel on a table. Now the table may be little bit tilted. So what will happen? This well may have like may start from here and this well may start from here. Means already we have a gradient. Since the identification, the like identification of the protein is based on what molecular weight in comparison with a
Yes, sir.
standard molecular weight. We call it marker.
Yes, sir.
Marker. Since the comparison is with
Marker.
the
Yes, sir.
marker, if there is difference in little bit of migration, then definitely there will be wrong identification may be there. So to minimize that, what we did,
Okay, sir.
at first we created say around one third of it we created, less than that we created as a stacking gel. Wherever whatever may be the depth of the well, all samples will reach at the top of the resolving gel at same time. Okay,
Okay, sir.
that is the function of the stacking gel. Second is there is pH difference. The stacking gel is having acidic pH, whereas the resolving gel is having basic pH. And
Basic pitch. Okay, sir.
there is a reason because in buffer we use tris glycine. So tris glycine is having say low charge, or we can say it is low dissociation at acidic pH, but the moment it enter in the basic pH, it becomes fully charged. So in that case, what happens that it helps in separation. Okay.
Okay, sir.
So in resolving gel, we get the separation. Okay.
Okay, sir.
Okay then. So I'm stopping here today,
Okay, sir.
and then I'll share with you uh, the one presentation of the remaining part, and that we will try to uh, like finish in our two classes. We are left with two classes, so
Yes,
we
sir.
will finish that portion. Now I am having some like no immunological and radioisotopic techniques.